Okay, so I'm tempted to say that I'm going to present a modeling presentation and not show you any models. Can't remember all my slides, but I think that's true. What I want to do is take us through. We've seen a lot of very good modeling efforts here. What I want to look at, and it's my role at Dow, is to apply the theoretical that comes out of these models to the realities on the ground. I work in science policy, so I'm working with regulators, I'm working with governments, I'm working with growers. What do these models really mean? We're all familiar, I think. This is a complex system that we're modeling, many moving parts, how we use the products, the genetics of resistance, and then the pest biology, and we've seen a lot of parameters today that relate to these things, but particularly around adoption, refuge implementation, cropping system and diversity, field and farm layout. We haven't really touched on those yet, so I want to try and introduce some of that as we go through this presentation. We've also seen a whole range in terms of the complexity of the models that people are playing with. You've got Dave Onstad, who's very complex. I don't know who I'm going to call simple, but maybe me. We have this huge range in terms of the complexity of the models and how they relate to the real world. So the simple models are descriptive, deterministic, lacking population dynamics, abstract in terms of space. They're assuming a constant environment. And you get a generalized model that will give you generalized solutions. It'll give you the big three parameters. And then you get the Onstad complexity, more mechanistic, more real biology in there. We're introducing stochasticity, we're introducing population dynamics, spatial processes, and we can even go on and add weather and so forth. And these become very system specific. We have a corn rootworm model for Illinois, and then we have a separate corn earworm model for North Carolina, and then a separate Spodoptera model for Brazil. And these all teach us different things. Simple two-patch model, I think this is what Ben was working with, where you got two areas, one of BT crop and one of refuge. You got two genes. We're assuming that resistance is monogenic. We're assuming that there's therefore three genotypes. We're assuming there's panmixis, that they go into a big pot, or I've got them going into a big cloud. And then they redistribute across the landscape. We assume it's random, maybe it's not. Generational time steps, we tend to assume discrete generations because it's easier. Deterministic. We're assuming uniform selection pressure. We're assuming that what happens in generation 1 also happens in generations 2 and on for 1,500 generations, or however long you can leave the model running. We're assuming 100% adoption of the technology, so every farmer is doing exactly the same thing, and 100% compliance with the refuge piece. So I look at these models, and when I talk about them within the company, I introduce it by saying, what I'm going to tell you is wrong, because that's too simple. But it may be wrong, but it's been extremely informative, and it has set up a resistance management paradigm in the US and Canada and Australia that we're trying to fit in around in other regions in the world, where we've got the big three factors determining the rate of resistance evolution: functional dominance, selection pressure and efficacy, and then refuge. Everybody throws up the high-dose refuge as being, the resistance management strategy, but really this is the special case. I think we can count on the fingers of one hand the number of high-dose refuge or high-dose crops, high-dose pest combinations that are out there in the real world today. We've got European corn borer, we've got tobacco budworm in cotton, we've got pink bollworm in cotton, so I don't even need a whole hand. but it's a really powerful resistance management strategy because you are controlling resistance. You're taking resistance alleles out of the population, which with a low dose product, you're not taking resistance alleles out of the population. So the refuge doesn't just dilute selection pressure, but the BT crop is removing those resistance alleles. So you get this huge bang for your buck. You can plant a small refuge and get a huge extension in the time until resistance develops. So we learned all that from those simple models. And then we started to ask more interesting questions. And these are the questions I think we've been talking through this morning. Insect behavior, dispersal behavior. So this panmixis is not true. Exactly how does mating and oviposition affect things? So the effective refuge size could be increased if the dispersal from the refuge is low. Or the effective refuge size, as we've just heard, could be decreased if insects avoid it. The high-dose refuge approach is enhanced if dispersal from the BT crop is high and you're not just recycling your resistant insects through the BT crop. Resistance is delayed longer if oviposition is biased towards the refuge, or it's the reverse of what Ben just told us. Density-dependent mortality can reduce the effective refuge size, and insecticides on the refuge can reduce the effective refuge size as well. These are all things that are happening in the real world, and these are all things that we would love to ignore in our models because they create too much complexity. So then we do end up adding these complex, and they become, as I say, very system-specific models with all these processes that we missed out from the initial models. Stochasticity, dispersal, you've got daily dispersal versus seasonal dispersal, you've got local host finding and mate finding versus true ecological dispersal, so within fields, among fields, across a landscape. We can start looking at adoption of the technology, we can look at refuge compliance, and we can reflect the reality that it's not just a single toxin out there today. Back in 1996, when we all started doing this, there was Cry1AB in corn, and it's basically the same thing as Cry1AC in cotton. Today we have a wider range, not a huge range, but a wider range of toxins that are out in the landscape at the same time, and they're all doing different things. Multiple resistance alleles will go with those multiple different toxins, and then the way that you present those in the environment, whether as pyramids combined in one plant or as a mosaic of different plants, and there's obviously combinations of those. We've got the refuge in the bag, the blended refuge that David talked about. We can start playing around with field shape. We're all assuming that fields are squares. I'll show you in a minute, they're not. Fitness costs, we've talked about that. Cross-resistance among these different toxins, and what does cross-resistance look like? How do heterozygotes that are resistance allele for one and some cross-resistance to the other, is that cross-resistance dominant or recessive, and so forth. There's lots of things that we don't do. One of the early things that we did do was add space. So these spatial models, again, we assume that a field is square and that they're laid out in a grid because that's the easiest way to program a computer, not because that's the way the real world looks. So in this case, this is a two-crop system, corn and cotton, and we've got some refuge fields. And you can run the model and you see what does that do to the resistance allele frequencies across that landscape. And you can get these localized hotspots of resistance where it starts to develop and then spreads out through the population. But the real world is not a system of grids. This is a satellite photograph of a farm in Illinois, I think it is, where there is a square field here, but there's a row of trees here. How's that going to affect dispersal between these fields? There's a creek running through it somewhere. There's a house, the farm buildings. They're all excluded from our model, and they're all going to affect the behavior of the insect as it moves through that landscape. And then that's just a little part of the landscape. For a lot of these insects, the landscape is hundreds of miles. So this is a little field, it's Indiana, sorry, not in Illinois. But this insect is affected by what's going on in Indiana, Illinois, Tennessee, Mississippi, and some other states I can't remember. With different crops, you've got non-crop areas, you've got pasture, you've got forests. All of these, the insect has to fly through these areas if it's going to be moving around. So we have all this complexity out there that most models omit. So truly looking at the insect biology, development rates, larval behavior, toxin avoidance of larvae as well as adults, mating preference and so forth. Realistic population dynamics, I haven't seen a really good like a predator prey or a parasitoid host model with genetics of resistance thrown in. So density dependence, density independence, quantitative genetics. We all assume it's one gene, one allele. That allele is giving you complete resistance. And then G by E effects on dominance. So we're measuring dominance. I pulled out a dominance value from my work on Spodoptera. But that may be in the lab and not in the field, and it may be in some fields, and it may be different in other fields, and maybe different in Puerto Rico, from Cuba, from Brazil. Abiotic factors. We're assuming, again, that uniformity across our landscapes, but we know that weather is real, and we know that soil types are different, and we know that those affect how insects do. Same with landscapes. It's way more complex than we're modeling. Farmer behavior. Again, we're assuming some uniformity in terms of what farmers are doing, not just with compliance with the refuges, but how they're managing their pests, what other things they're doing on their farm to manage their crops. Crop rotation, insecticide use. Tillage is a big question. What effect does tillage have on insect populations, and how has the adoption of herbicide-tolerant crops reduced tillage, which may allow more insects to survive, and if those insects are in the refuge or in the BT, and we don't tackle these things. Cropping diversity and introduction of new technologies. We're assuming, again, we've released Cry1AB corn in 1996, and then we assume that that's static and it's going to be Cry1AB corn for the next 150 years and nothing else. And we know that well, we hope that's not true, because it's not my product. And then there are longer-term changes in agriculture. I looked at this same area that I showed you on the previous slide, that maybe a realistic scenario for a corn earworm or a fall armyworm moving around. We assume in our models that you put corn in that field, that's a cornfield, but it's not. We look at what was planted as cotton in, which way around is this? I remember the slide now. So a field that was corn in 2001, was it still corn in 2010 or 11, whatever this is? Same for cotton, same for soybean. And no, the crops in each field is changing. If you switch out a cornfield if you plant cotton in your cornfield, you don't have your corn pest to worry about anymore, and they've had to go somewhere else, and we need to include that in the model. And over that same time period, there's been a shift in the relative amounts of cotton, soybean, and corn in the landscape. Cotton production has declined over the last 10 years. Soybean has increased, and corn has increased. But again, our models are assuming that these things are all static. And then, again, the changes in the traits that are available through time. So we started out in the mid-90s with one gene, one crop. But we've introduced new crops through time, new genes through time, and we've started stacking them, creating pyramids. And so our program in 1996 to make Cry1AB last for 15 to 20 years got superseded by the technology as we replaced it with new and better things. So that 15 to 20 years was actually perhaps not the right goal that we should have been going for. And of course, as every model in this room knows, we can do things in the virtual world that we can't do in the real world. So controlling some of these things that I've talked about as being important, and we've heard. So controlling insects dispersal. We can assume the spatial uniformity and temporal stasis. We can fix parameters that we know are inherently variable. How many of the insect modelers here use a single value for fecundity? Oh, okay. Only Dave. Oh, thank you, Mike. And how many insects lay exactly 200 eggs? So we know that our models are not real. They're not the real world, and we can't guarantee farmer behavior. I can talk to you about that for hours. But that doesn't mean that they're not useful. They have to simplify the real world, or we will never understand what's going on. So these models they are useful learning tools, education, research tools. They are not predictive. Somebody asked earlier, can we make these models more predictive? Maybe we can with enough knowledge and enough computer power and enough time. But is that really the goal? Do we want to know that if I release this product and nothing else changes for the next 20 years, would it still work? Or do we want to know, if I release this product, what are the better strategies that I can use to increase the likelihood that it'll still work for 20 years? And that's really where we need to go with the way we think about these models for resistance management. And it's very easy for me to be sitting in my office to dream up these fantastic schemes for what growers should do, what regulators should do, what a research entomologist should be doing to manage resistance. But how many of those are really practical? So farmers are not in the business of raising susceptible bugs. Farmers are in the business of raising a crop, feeding the world today, and hopefully putting in strategies that will enable them to keep doing that for the next 20, 30, 40 years. But we have to balance those short-term costs that resistance management brings with the long-term benefits. And where that balance falls is not a science question, it's not a modeling question. It's a policy question, it's a crystal ball gazing question. What will be available, what new technologies will come along, and so forth. So I'm constantly asked within the company, how much refuge size do I need for BT soybean in Brazil? Can you run your model and tell me how much refuge size I need? And my answer is no, I cannot, because it's not a scientific question. It's how long is a piece of string question. What I can do is use the model to indicate what are the relative risks and benefits of a 20% refuge versus a 10% refuge, of a refuge in the bag versus a structured refuge. We can look at what's the worst case that could happen. We can look at some more realistic cases. That's how the models should be used. That's how we need them to help us in our decision making as the developers of these products and as regulators, to help us do the best that we can to achieve that balance, whatever that balance is between the long term and the short term. So we as modelers all have a responsibility to do this in a transparent way, and to be honest about what our models can and cannot do in the decision-making process. So we have some responsibilities. First, obviously, get the code right, get the math right, and get the code right. Hopefully, that's the easy bit. I think one of the speakers, I can't remember who it was, maybe it was you, Nina, talked about setting out what is the goal of my modeling project before you even put pen to paper or start typing code. What is it? What is the question that I want to answer? And create your model to test that hypothesis. Just like doing any other experiment in the field, you've got to have your hypothesis, otherwise your experiment is not informative. You need to relate the model endpoints to the real world. So 0.5 has been bandied around as being a good threshold for resistance failure and no resistance success. That may be true in some situations. It may not be true in other situations. We've all used different endpoints during our careers as modelers. Is your definition of resistance based purely on the gene frequencies? Or do you factor in population size as well? When your product is failing, you've got a larger population. Got to clearly state all your assumptions, both in the model structure and in the parameterization. I can't count the number of times that I've sat down with a model and we've come out with two very different conclusions about ostensibly the same question. And we'd look, what's wrong with the model? How do our models differ? But it's not. It's the parameterization, dispersal, genetics of resistance. Those are what drive these things. So we have to be very transparent with those. We have to recognize that these models are simplifications of the real world. We have to understand the dependencies within the model that would, if we change those dependencies, would alter the conclusions. So the sensitivity analyses, they need to be more complex than what we're doing. And then we need to be better communicators. So again, how do we apply our models to the real world? How do we use them for decision making? It's not we cannot spit out the right refuge size just based on a model. And then uncertainty, and I think we'll talk about that later today. So that being said, models have served us well. We've learned some very strong and very clear principles of resistance management using models, be they simple or complex. The refuges reduce selection pressure. The high dose takes out resistance alleles, very powerful strategy. Functional dominance is driving the single gene outcomes. We've seen that pyramids are more durable than single traits. And we're learning that if situations where compliance with structured refuge is low, it's better to put that refuge in the bag, use a seed blend. And our experience over the last 15 years has really supported that what we've learned from the models is true. The exceptions to expectation actually prove the rules in this case. So my first model when I was at NC State was Helicoverpazea, corn earworm in a corn-cotton ecosystem, moving between the two crops multiple generations a year, flying around between fields and so forth. Predicted that we would get resistance in 5 to 8 to 10 years. Here we are 15 years later, we don't have resistance to BT crops in corn earworm. And why not? Because we replace those single gene products with 2 gene pyramids, and we've recognized now that natural refuge is more important than any structured refuge that the farmers were told to put out there. The same with Spodoptera in Puerto Rico, resistance to Cry1F. Oh my God, it evolved in 3 years. That's exactly what Fred Gould said back in 1986 would happen with these products. But what happened to our fantastic resistance management strategy? Well, that 3 years was actually 30 generations of the insect. That's not what Fred Gould said. So there was some mitigation of the resistance of the selection there. Refuges were planted, but they were lost through drought and through pest pressure. The fall armyworm came and ate up the refuge. There was no refuge left for the insects to lay eggs in, even if they weren't repelled from it. So it was the refuge strategy was not a durable strategy there. And then there was the geography of Puerto Rico with the mountains and the ocean is very different from the Midwest that everybody was thinking about before. So as we move from the virtual world to the reality world, what are the questions that are still out there for us? And I think this workshop hopefully will move us towards this. And I'm really excited to have people in the room who are not entomologists, who have not been doing IRM modeling all their careers, but can bring some new ideas and new thoughts. The economics of resistance management is hugely important. We're seeing this with weeds, glyphosate-resistant weeds right now. They are causing huge economic problems to farmers, but they never took steps ahead of time to try to delay resistance. And we know resistance management strategies have a short-term economic cost. So then there has to be a balance between that short-term economic loss and the long-term benefit And if you plant a 50% refuge, do you still get that the overall economic benefit of the technology? Do you double the durability of the technology to pay back that 50% refuge? Sometimes. What remedial actions are effective in the real world? We have resistance situations out there now. There's a lot of talk around remediation. What are we going to do about this? Can models be used to help us understand how we could influence the gene frequency down, or at least influence the impact of the gene on the economic ability of a grower to raise a crop? IPM practices, everybody's talking again about IPM because IRM has failed. Integrated pest management. What are the other tools that farmers use, can use, should use to manage their populations, and how do those affect resistance management? And it's not just management of the target pests that we're concerned about for resistance, but management of all of the pests in their system, and even other all the agronomic practices going on. How do those affect resistance alleles, resistance genes? What about uncertainty? We all know that our models are wrong, and we know that there's a huge uncertainty. How do we factor that into our decision-making process? How does uncertainty change what we might do in terms of resistance management strategies? And then what about complex genetics? Incomplete resistance. Most cases of resistance out there today don't give resistant homozygous insects full fitness on the BT corn. How does that affect their evolution? Fitness costs, we've had some talk about that. About quantitative traits, we love to model single gene situations because the code is easy and the genetics is easy, and there's a whole field of evolutionary biology that is based on quantitative traits. None of us have looked at that from a resistance standpoint. We need that. I don't have the mathematics to do that. And even just having more than one allele involved, one allele per locus. So we've got one resistance gene maybe, but you've got competing alleles at that gene. And we know there are multiple alleles for the cadherin mutations that cause resistance in pink bollworm. I haven't seen a model that's incorporated those and looked at different frequencies of those. I think that was it. So there's some thoughts on where we could go and what groups we could form, Nick. Okay.